In parts of Asia, for certain pathogens, resistance levels that were over 50% have dropped significantly, down to around 22% over a couple of decades. That's a direct result of applying comprehensive risk mitigation strategies.
So concerted effort really does pay off.
It absolutely demonstrates that proactive, multi-pronged strategies can work.
A whole new era of communication in the global aquaculture industry is coming. Now you have the brightest minds in aquaculture right in your pocket. And what's best, you can listen to all of them while driving to a farm, traveling, or running errands. It's never been this good, and it's never been this simple. Welcome to the Aquaculture Podcast Show, the first AI-based podcast in aquaculture, where you'll find cutting-edge insights in everything that's working in aquaculture, nutrition, health, and production.
Welcome to the deep dive.
Okay, today we're plunging into a topic that's just, well, absolutely critical for the future of aquaculture. Antimicrobial resistance, AMR.
Indeed.
Now, if you're in this industry, you already know aquaculture is, frankly, a global powerhouse. It provides more than half the world's seafood now.
An incredible achievement.
Totally. It's a real testament to innovation, efficiency, delivering vital protein.
Yeah.
But alongside this amazing growth, there's this complex, growing challenge. Antimicrobial resistance. It really demands our focus.
It absolutely does.
So our mission here today is to pull out the most critical insights on AMR and aquaculture. We want to look at the scale, how resistance actually spreads, and what cutting-edge strategies are being used to tackle this global threat.
Getting straight to the point.
Exactly. Getting you the core knowledge you need on this.
To really get a handle on this, we have to see AMR in aquaculture through that One Health lens. It's fundamental.
Right, that connection between everything. Exactly. This isn't just a fish farm problem isolated over there. It's deeply, unavoidably interconnected. Human health, animal health, environmental health.
It all links up.
It all links up.
Yeah.
What happens in the water and the fish, it doesn't stay contained. It directly impacts public health, the wider ecosystems we all depend on. That interconnectedness is, well, it's central to grasping the full picture.
That One Health framework is definitely key. So, okay, practically speaking then, in aquaculture itself, where did the consequences of antimicrobial use first really show up?
Well, it starts with why they're used in the first place, right?
Yeah.
To prevent and treat bacterial infections in fish.
Which is crucial for healthy stocks.
It's absolutely crucial,
Yeah.
especially more intensive systems. But here's the rub. It's the misuse. Incorrect dosing, perhaps, treatment for a wrong duration, sometimes just unnecessary use altogether.
Right.
That improper use directly speeds up the emergence and, importantly, the spread of resistant bacteria. It's like we're accidentally accelerating evolution against ourselves.
Wisenetics turns podcast airtime into brand authority. We don't sell ads, we elevate voices. Curious how far your voice can go to become a reference in the industry and attract more leads? Scan the QR code and discover how we can turn your expertise into unmatched brand authority. Let's transform expertise into influence, starting now.
If these resistant bacteria are building up in the fish or the farm environment, how direct is that risk to people? Does it actually reach our plates?
Oh, it absolutely can.
Yeah.
The link to human health is direct. These resistant bacteria aren't confined. They can transfer to humans, particularly through eating aquatic products, and especially things consumed raw or, undercooked.
Like sushi, smoked fish, that kind of thing.
Exactly. Sushi, smoked fish, popular items. That creates a direct route for these resistant strains into the human food chain.
Yeah.
And that's a very real, very critical public health concern.
So not just what we eat, what about the wider environmental impact? You mentioned ecosystems earlier.
Yeah, the environmental side is... It's huge.
Yeah.
Here's a pretty stark figure. Around 75% of the antimicrobials given to fish, they're released largely unaltered into the surrounding environment.
75% just released.
Pretty much unchanged chemically. So we're effectively seeding resistance genes across aquatic ecosystems. We find resistant bacteria, antimicrobial residues, not just in the farm water.
But further out.
Much further. In the sediment, in other aquatic organisms that weren't even the target. The environmental footprint is way bigger than you might initially think.
Okay, so serious consequences. Let's unpack the drivers then. What's actually pushing this resistance in aquaculture?
Well, a primary driver, no doubt, is the overuse and misuse we talked about. It's still prevalent.
And often without enough vet oversight.
That's often a factor, yes. It's a challenge amplified by the sheer scale and global nature of aquaculture.
Yeah.
And what's Particularly worrying is the continued use of certain antibiotic classes that are critically important in human medicine.
Which ones are we talking about?
Things like quinolones, tetracyclines, sulfonamides, and phenicals. Broad spectrum, powerful tools we really need for serious human infections.
And they're used commonly in fish farming.
Widely employed, yeah.
Yeah.
And then adding another layer, in some parts of the world, these compounds might also be used off-label as growth promoters.
Ah, right. Not even for treatment.
Exactly. Which just adds to the selection pressure for resistance.
That is a critical point. So, okay, these resistant bacteria emerge. How do they spread so effectively? What are the mechanics?
It's quite fascinating, actually. It's not just slow, gradual mutation within a lineage, though that happens vertical gene transfer, parent to offspring.
Okay.
But the really rapid spread, that's facilitated by something called horizontal gene transfer, or HGT. Bacteria literally share resistance genes directly between each other.
Like trading blueprints.
Almost exactly like that.
Yeah.
They can do it through direct contact conjugation. They can pick up free-floating DNA from the environment, that's transformation, or even use viruses, bacteriophages, as sort of couriers, that's transduction.
Wow. So multiple routes for sharing resistance.
Multiple and highly efficient routes. And this brings us back to the water environment.
Why is that environment so special for this?
Because aquatic environments are almost uniquely suited for these genetic exchanges. You often have incredibly high densities of microbes packed together.
Lots of neighbors.
Lots of neighbors. And frequently, existing low levels of antibiotic residues already present in the water or sediment.
Creating a sort of hot zone.
Precisely, a microbial melting pot. It's an ideal breeding ground where these resistance genes can swap, multiply, and spread really efficiently across different bacterial species.
So it's more than just the farm itself. Are there other external factors feeding into this environmental spread?
Oh, definitely. Wider environmental factors really amplify the dissemination. Think about wastewater discharge from the farms themselves.
Carrying resistant bugs and maybe drug residue?
Both, potentially. Then there's agricultural runoff from land farming that can carry residues from livestock use. Industrial pollution can sometimes introduce compounds that inadvertently select for resistance too. And even the feed, if it's not sourced and handled carefully, could potentially introduce resistant bacteria into a system. All these pathways contribute to a bigger environmental load.
It's a complex web.
It is. And add to that biofilms. These communities of bacteria that form on surfaces, pipes, tanks, equipment.
Like a protective slime layer?
Essentially, yes. And they act as real refuges for resistant bacteria. Inside a biofilm, they're more protected from environmental stress, and actually, genetic exchange, like HGT, is enhanced within that structure.
So they become persistent reservoirs of resistance.
Exactly. Persistent sources. And then, stepping back even further, we have to consider things like climate change. Rising water temperatures, that could potentially speed up bacterial mutation rates, change pathogen dynamics. It adds another layer of complexity to control efforts.
This is incredibly complex, which leads to a crucial question. How well are we actually measuring all this? You can't really manage what you don't understand properly.
That's a really key point. Current monitoring, especially in some regions, often isn't adequate, particularly where there aren't strong, integrated national AMR action plans in place.
So how do we detect it? What are the methods?
Well, you have your standard lab methods like disc diffusion or broth dilution. They give you a basic picture of resistance patterns, foundational stuff.
But maybe not the whole story.
Not the whole genetic story, no. For that, you need the more advanced molecular techniques. Things like real-time PCR to look for specific resistance genes, or even whole genome sequencing, WGS.
Giving you the full blueprint.
Exactly. WGS lets you track antimicrobial resistance genes, understand how things are being transmitted, really map out the dynamics. That level of detail is crucial for informing targeted, effective strategies.
But is it straightforward to interpret that data?
Wow. Well, that's where we hit a significant barrier, especially for bacteria common in aquaculture. We often lack clearly defined red lines.
What are lines?
Yeah, what scientists call clinical breakpoints or epidemiological cutoff values, ECVs. These are standardized thresholds that tell you if a level of resistance is likely to cause treatment failure or if it's unusually high for that bacterial species.
And we don't have those for key aquaculture pathogens.
For many important ones, like common species of Aeromonas, Vibrio, Streptococcus, Edwardsiella, these breakpoints, or ECVs, are either missing or not well established for the aquatic context.
So it's hard to know how worried we should be about a specific finding.
Exactly. It's like trying to gauge a fever without knowing what a normal temperature is.
Yeah.
We urgently need more research to establish these reference points. That would dramatically improve our ability to accurately assess the risk and respond effectively.
Okay, so given this complex picture and the measurement challenges, let's pivot to solutions. It seems obvious we can't just keep using antimicrobials the same way. Alternatives must be a priority.
Absolute paramount. The shift towards alternatives is critical, and thankfully there's a lot of promising work happening.
Like what?
Well, for boosting fish health directly, you've got things like probiotics, beneficial bacteria for the gut, prebiotics compounds that feed those good bacteria, and immunostimulant substances that directly kickstart the fish's own immune system.
Strengthening the fish from within.
Precisely. Then there are natural compounds showing promise, like certain essential oils or antimicrobial peptides. And a really exciting area is phage therapy.
Using viruses to fight bacteria.
Exactly. Bacteriophages are viruses that target and kill specific bacteria, but they're harmless to the fish and other beneficial microbes. Very targeted control. And of course, we can't forget vaccination.
It's still a cornerstone.
Absolutely. Vaccines against major pathogens like Aeromonas salmonacida, which causes furunculosis, or Vibrio anguillorum, have been huge successes, drastically cutting the need for antibiotics in some sectors.
These alternatives sound great, but they surely need the right conditions to work effectively. They're not magic bullets.
Oh, definitely not. They have to go hand in hand with really solid husbandry practices. It's all about creating an environment where disease is less likely to occur in the first place.
Basics matter.
The basics are fundamental. Optimal stocking densities, not overcrowding. Robust biosecurity measures preventing pathogens from getting onto the farm. And actively minimizing stress for the fish. Healthy, unstressed fish are naturally more resistant. These practices reduce the initial disease burden, which reduces the need for intervention. So building on that, you need that truly holistic One Health approach to mitigation we mentioned earlier.
Bringing it all together.
Exactly. Enhanced biosecurity and hygiene right across the farm. Regular disinfection, controlled access, proper waste management containing potential contamination.
Water quality, too.
Critical. Rigorous water quality monitoring, keeping parameters like pH, oxygen, ammonia in the optimal range reduces stress and susceptibility. Farmer training is also huge, making sure everyone understands responsible antimicrobial use.
Empowering the people on the ground.
Yes. And longer term, genetic selection plays a role. Breeding fish strains that are naturally more resistant to specific diseases. We've seen success with rainbow trout resistant to bacterial cold water disease, for example.
Smart breeding.
And finally, you need strong surveillance and regulation, tracking antimicrobial use, monitoring resistance trends consistently. That data guides policy and keeps the industry accountable.
Now, after laying all this out, it might sound a bit daunting, but there is good news here, right? We are seeing some progress.
Yes, definitely. And that's really encouraging. If you look at the global usage figures, they're substantial, around 10,000 tons in 2017, maybe projected to rise towards 13,600 tons by 2030. And the Asia-Pacific region uses the vast majority, nearly 94%.
Okay, so usage is high.
Usage is high. But, and this is the crucial part, where integrated AMR control policies have been properly implemented, we've seen real measurable reductions in resistance levels.
Can you give an example?
Sure. In parts of Asia, for certain pathogens, resistance levels that were over 50% have dropped significantly, down to around 22% over a couple of decades. That's a direct result of applying comprehensive risk mitigation strategies.
So concerted effort really does pay off.
It absolutely demonstrates that proactive, multi-pronged strategies can work. They yield results.
So wrapping this up, what's the big takeaway? Our deep dive really shows AMR in aquaculture is, well, it's complex, multifaceted, deeply tied into global health, environmental health.
I think the central message that comes through so clearly is this. If aquaculture is going to continue expanding sustainably, which we need it to, it must integrate environmental stewardship, genuinely prudent antimicrobial use, and strong collaboration across sectors, all under that One Health umbrella.
It's essential for the industry's future health and credibility.
Absolutely. It's about ensuring this vital food source thrives responsibly without adding to the global AMR crisis.
Indeed. Which leaves us with a final thought for you, the listener, to consider. Given this interconnectedness and seeing the proactive steps and the real progress aquaculture is making in tackling AMR, what broader lessons might other agricultural sectors learn from aquaculture's approach as they face their own antimicrobial challenges?
That's a great question. There's certainly potential for cross-sector learning there.
Something to mull over. Thanks for joining us on The Deep Dive.
If you enjoyed this episode, make sure to follow the Aquaculture Podcast Show on your favorite platform. And don't forget to follow us on LinkedIn, Instagram, and Facebook to stay updated on the latest episodes and industry insights. Thanks for tuning in, and we'll catch you next time.